Today we are going to discuss about cell, the unit of life, especially about prokaryotic and eukaryotic cells. So first I will be explaining about prokaryotic and eukaryotic cells. Then we will be discussing about the detailed difference between prokaryotic and eukaryotic cells. So the cell is the unit of life. So basically it is the fundamental structural and functional unit of all our living organisms. So they are basically of two types. One is prokaryotic cells and the other type is eukaryotic cells. Now prokaryotic cells. So this is the structure of the prokaryotic cell, wherein we have many kinds of in the detailed cell structures we can observe the parts like nucleoid region is the red color one, chromosome, DNA, villi, flagellum, ribosomes, cytoplasm, cell wall, plasma membrane. So now we will be discussing about each one of them in detail. So moving on to the prokaryotic cells. So the word pro itself indicates that they are first initially discovered. Now carrion here indicates carrion indicates nucleus. So these cells were first discovered, so that is the reason carrion means nucleus. So that is the reason they are given the name prokaryotic cells. Now let us understand about them. So these prokaryotic cells are generally smaller and multiply more rapidly than the eukaryotic cells. They basically vary in their shape, size, and have no membrane-bound organelles such as your nucleus and other organelles. So these basically include bacteria, blue-green algae, which are also known as cyanobacteria. Blue-green algae, we also call them as cyanobacteria. Next is the PPLO, that is pleuropneumonia-like organisms. So this is the structure of bacteria. This is the cyanobacteria, which I have just said the blue-green algae are present in this. Next coming to this is mycoplasma, that is very small and minute sized organelles. Now let's discuss about bacteria. So bacteria have basically four shapes. Like in the nature they are present in these four basic shapes, that is bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, spirillum, spiral. So here we can very clearly observe this. Bacillus is like in the shape of a rod, whereas coccus is it in the form of spherical round shape. Vibrio is nothing but comma shape. So whenever there is any kind of comma shaped bacteria, that we will call it as vibrio. So here we can see it is in the form of comma like this. Next is the spirillum, like this. They are rounded and roll structures, so they are known as spirillum. So here if we come to the detailed discussion about their shapes, here we have spheres, that is streptococci, diplococci. So based upon the number of cocci which are attached here, we have got their names as, if it is a tetrad, then it is streptococcus. If it is a two spherical spheres form, then it is diplococci. Then we have the staphylococci, wherein we have many more than 4 number of spherical cells attached with each other. Next is the sarcina. Coming to rods bacilli. So here it is a chain of bacillus. Best example is Bacillus anthracis. Next we have the flagellus rods. Salmonella typhi, which causes typhoid, is also bacilli, which is having a rod shaped bacteria. Next we have your Clostridium botulinum, which causes food infections, so it is also a rod shaped bacilli structure. Next we have spiral shaped Vibrio cholera, which causes cholera. So it is in this spirulum form, in this kind of structure, spirulum structure. Next Helicobacter pylori, which we see in our stomach, commonly known as H. pylori. Next we have spirochetes, that is Treponema palladium. Here also we have the structures of few bacteria. So Vibrio cholerae, as I've just said, this is the structure of Vibrio cholerae, which causes cholera. Next is the Salmonella typhi, which causes typhoid. This is Clostridium botulinum, which causes food food contamination, that is through this botulism is found in food, and then we can find this kind of bacteria in the food which causes food contamination. So primarily it is concerned with food contamination, Helicobacter pylori, which is present in our stomach. So this is all detailed information about bacteria. So moving on to the next part, we will be discussing now actually what are the different kind of cell organelles that are present in the prokaryotic cells. So here we basically have 7 cell organelles, that is cell envelope, mesosome and chromatophores, nucleoid, flagella, pili and fimbriae, ribosomes, inclusion bodies. Now if we observe, this is the structure of the prokaryotic cell, wherein we have, this is the structure wherein we have, see, this layer is the plasma membrane, the yellow color layer is the cell wall, and the outermost layer is the capsule. This blue color portion is the plasmid. These finger-like out projections are known as pili, and these red dot-like granules are known as ribosomes, and the spiral round part, which is called in nature, is known as nucleoid, that is nothing but our DNA, deoxyribonucleic acid. Next, this tail-like structure, this one is known as flagellum, and all these organelles are emerged in this known in a blue color liquid, which we are able to see here, that is known as, it is the medium in which all these organelles are being present, that is the cytoplasm. Now we shall discuss about each one of them in detail. So first, coming to cell envelope. So as I have just said, if we observe this structure very carefully, here we have 2 and 3 layers. So first one denotes the glycocalyx, and the second layer, which is in dark yellow color, denotes the cell wall, and the third one, which is having green color here, denotes the plasma membrane. So as I have just discussed, this liquid is cytoplasm, and the remaining structures, this is the DNA, then these hair-like projections are pili, and this tail-like structure is your bacterial flagellum, ribosomes and plasmid. So chemically we have our cell envelope, which is a protective covering. So this is the first primary outermost covering, the cell envelope. This is the primary outermost covering, which is this is the outermost covering, which is more helpful for the cell wall in protection. So here we have basically 3 layers, that is the glycocalyx, cell wall and plasma membrane. So 3 tightly bound layers. Coming to the very first layer here, this is the glycocalyx. It is the outermost layer, and its composition and thickness vary in different kinds of bacteria. That is purely dependent upon the bacteria size, shape, and it is a slimy layer. So this word slimy layer is nothing but it is a loose sheath or capsule which is thick and tough. So due to its slippery nature, it's like gel like. So that is the reason we call it as slimy layer, slime layer or slimy layer. Next, coming to next organelle, that is the cell wall. So the second one, this yellow color layer, is the cell wall, which is the middle layer, which is seen in all the prokaryotes. This is mainly responsible for the shape of the cell and support for the bacterium by preventing the bacterium from bursting or collapsing. So during unfavorable conditions, this cell wall helps the prokaryotes by supporting them in order to prevent the bacteria from bursting or collapsing. Coming to the last innermost layer, that is the plasma membrane. So if we observe this green color one, so that is the reason I have given the color denotions in the same way. So here this red color layer, that is the first layer, glycocalyx, first one. Next, the innermost one, it is in yellow color. I have given the same color for the cell wall. This is the second layer, and the plasma membrane is the last one, that is the third one. So that is the reason I have given same green color for easy understanding. So this is the innermost layer, and it is semi-permeable in nature. So here the concept of permeability comes. If the molecules are able to enter from one membrane to another membrane, permeability, if the molecules are able to enter from one membrane to another membrane through without any obstruction. So that is the concept of permeability. So here it is semi-permeable, means only few ions or few molecules can enter into the layer. Few molecules or ions can enter into the layer, and that interacts with the outside. So this is almost structurally similar to that of eukaryotic cell. So the permeability, as it is the innermost layer, it is semi-permeable. So only few molecules can enter into that. Not all molecules. Again, we have the concept of permeability, hydrophilic and hydrophobic. Hydrophobic are water resistant molecules, hydrophilic are water favorable molecules. So this acts as a barrier for the entry into the innermost part of the cell wall by protecting the cell wall in the adverse conditions. Now moving on to the next part of cell envelope. Now why is this semi-permeability found? So basically based on the type of cell envelope and its response to Gram staining, we generally perform an experiment called Gram staining. In order to find out whether the bacteria is Gram positive bacteria or Gram negative bacteria. So this is the Gram staining experiment. So whenever we have taken a few Gram positive bacteria and Gram negative bacteria on a slide, and we have given this crystal violet treatment to that. So as soon as they are treated with that, both of them turn into violet color, and then again they are further treated with the iodine. So again they change their color. Then finally after some time there is decolorization in case of Gram negative bacteria, but Gram positive bacteria remain in the same color. So at the end, when we counter stain with saffron and dye, so here we use crystal violet dye first, and the last we will add the saffron and dye in order to finally find out which bacteria is Gram positive, which bacteria is Gram negative. So Gram negative bacteria will always have some color on it. So with that we can identify the bacteria is Gram negative, whereas these will always retain their color. So we can identify them as Gram positive. So that is what I have given here. They take up and retain the Gram stain. So since starting, if we observe here, we can easily understand the nature of Gram positive bacteria, whereas Gram negative bacteria in the decolorization step, they will not retain their color, they will lose their color, and ultimately they change into some other color apart from the Gram stain. So they do not retain this Gram stain. So if at all we observe the structural differences here in terms of Gram positive and Gram negative, there are many layers in gram-negative bacteria, so that is the reason they can't retain this color stain. But whereas in gram-positive bacteria, there are only 2 to 3 layers, so they often retain the crystal violet dye reaction here in the second step, and they stain dark violet. Whereas in gram-negative bacteria, they are always decolorized to accept the counter stain. So they have this pink color of saffron. So that is the identification done to identify what kind of bacteria it is, whether it is gram-positive or gram-negative. So moving on to the second cell organelle in prokaryotic cell, we have mesosome and chromatophores. So here in the structure of prokaryotic cell, these are, here we have mesosomes, this is the nucleoid, and inside that we have this chromatophores. Now mesosome is nothing but, here we can observe a blue colored structure which is having many number of foldings. So it is formed by the infoldings of plasma membrane. So plasma membrane, that is the third innermost layer which is present in the cell envelope, undergoes several kind of foldings to form a structure known as mesosomes, which includes vesicles, tubules and lamellae within it. So the main function of mesosome is cell wall formation and DNA, that is nothing but in chromosomal replication and for the distribution of the chromosomes to daughter cells, respiration and secretion to increase the surface area of plasma membrane and the enzymatic content also. So these are the functions of mesosome. Now moving on to chromatophores. The name itself indicates, if we break this into two parts, chromatophores. These are nothing but the pigment containing membranous infoldings in some prokaryotes. The best example for chromatophores is cyanobacteria. Chroma is nothing but related to colors. Chromatophores. So here if we observe these chromatophores, it is nothing but a pigment which is present in the chlorophyll. So here if we take the structure, this is the chromoplasm. Inside we have rounded structures, that is nothing but the centroplasm or the nucleoplasm. So this is all about chromatophores and mesosomes. Now moving on to the next cell organelles, we have nucleoid. So nucleoid is nothing but our DNA, which is having many kinds of foldings, which is especially in naked form and non-membranous, having genomic DNA with single chromosome or genetic material and protein. So DNA is nothing but, we always know that it is a genetic material. But the thing to be noted here is it is having a single chromosome and a protein. So many bacteria have a small circular DNA called plasmid. So we can observe the small circular DNA structures. So this is known as plasmid, which is outside the genome of DNA. So this is nucleoid, this is plasmid. Now the reason why we are differentiating them is it gives some unique phenotypic characters, for example like resistance to antibiotics to bacteria. For example, streptomycin, this is an example for bacteria species, this is an antibiotic. So this feature helps the bacteria for the resistance against streptomycin. Now moving on to the next part called flagellum. Flagellum is like, this is the hair-like outprojection. They are extensions from the cell towards the outer side. So these are thin filamentous extensions from the cell wall in the form of motile bacteria. Their number and arrangement are varied in different bacteria. So it is not the same in all bacteria. Basically, if you observe this detailed structure of flagellum, it has three parts in it. This is the filament, this is the hook, and this is the basal body. So filament is comprised of three parts. The initial one is the filament, next the middle part is the hook, and the ending part that is towards the base is the basal body. So it is the longest portion and extends from the cell surface to outside. So if we observe here, it is coming from the cell surface to outside. So this is the flagellum or flagella. Just a singular and plural forms of flagella and flagellum. Moving on to the next parts of villi and fimbriae. So here the hair-like out projections which are present towards the outer cell. These are known as villi. If they are helping in the reproduction process, so that is the reason we are calling them as sex pillars. So these are surface structures and they have no role in motility. Now what is this motility? Motility is nothing but movement of the cell. So whenever there is any kind of movement to the cell, it is happening through flagella. If we observe clearly, it is written here from the cell of motile bacteria. So it always moves with the help of flagella. This helps in motility. But these villi and fimbriae are just attachments to the cell and they do not have any role in case of movement. So these are elongated, villi are nothing but elongated tubular structures which are made up of a special protein called pillin. So the protein which is responsible for the reproduction is pillin. So that is the reason. So these are like tube flagella, and here we have a tube-like structure called sex pillars. Next, fimbriae are bristle-like fibers sprouting out of the cell. So these are fimbriae which are coming out from the cell, and the tube-like structures are sex pillars. Hair-like projections are fimbriae and the tube-like structures are villi. So these help to the attachment of bacteria to the host tissues. If at all we observe in case of mycorrhizae where there is a symbiotic association, these fimbriae will help the bacteria to attach to the host tissues. So that is the reason these two are associated with the motility, not associated with the motility, but the fimbriae help in attachment to the host tissues. Moving on to the next part of prokaryotic cells, that is the ribosomes. So here internally we have certain red dot-like structures or soft granule-like structures. So these granule-like structures are known as ribosomes, and their size is about 15 to 20 nanometers, and they are always made up of two subunits, that is 50S and 30S. The division is like, if we take a 70S ribosome, it is divided, it has two subparts in it, that is 50S and 30S. So 70S is the bigger part of ribosome, it is given in the picture as well. 30S and 50S are its subparts. So S here is nothing but this Swedberg unit, or we can also call it as sedimentation coefficient, which is a measure of density and size. Now moving on to the functions, these ribosomes are very much important for translation process, that is the protein synthesis. So if we observe this second picture, the larger subunits as well as the smaller subunits get attached to the ribosome and they are transcripted in the form of mRNA, and these are again attached to a polypeptide chain, and the process here it is written, direct translation happens. So several ribosomes may attach to a single mRNA to form a chain called as polyribosome or simply polysome. So these 70S will get breakdown into 50 and 30, and these will attach it to a single mRNA structure, and then they together form a chain like this, and this entire chain is known as polysome or polyribosomes. Now these ribosomes of a polysome will translate the mRNA to protein. So this the conversion of mRNA into proteins, this process is known as translation. That's why I have given in the brackets as protein synthesis. Synthesis of protein from mRNA is nothing but translation. So moving on to the next cell organelle, that is the inclusion bodies. So inclusion bodies are very much small and minute structures. These are the inclusion bodies which are present in this inside the cell. These are generally non-membranous, so that is the reason they are like very much into the inner layer of the cell. That is the reason they are not having any kind of membrane. Basically, these store reserve material which are seen freely in the cytoplasm. So they are that is the reason they are in many number and they are freely moving inside the cytoplasm, as they store only the reserve food material. Now, how is this reserve food material stored? Is like, for example, in the form of phosphate granules, synovial granules, glycogen granules, or gas vacuoles. So the most common form which we observe is glycogen and gas vacuoles. Gas vacuoles which are commonly found in blue-green and purple photosynthetic bacteria, nothing but our cyanobacteria. Cyanobacteria have these gas vacuoles which have their food stored in the form of gas vacuoles through inclusion bodies. So this is a brief overview of prokaryotic cell. Now let's move on to eukaryotic cells. So moving on to eukaryotic cells, and this is the structure where we have many cell organelles enclosed inside the eukaryotic cells. So the basic difference between eukaryotic and prokaryotic cell is they have a well organized membrane bound nucleus and organelles. So the presence of these membrane will give them clear compartmentalization of the cytoplasm. They have complex locomotory and cytoskeletal structures, and their genetic material is also organized into the chromosomes. Most of the times we will find them in the form of chromosomes only. So the examples for the eukaryotic cell are like a protist cell, a fungal cell, a plant cell, and an animal cell. So there are more number of cells which are having these kind of organized membrane bound nucleus and organelles. So moving on to the parts of cell organelles of eukaryotic cell. Here there are cell membranes, cell wall. We also have endomembrane system comprising of endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. We also have mitochondria, plastids, ribosomes, cytoskeleton, cilia and flagella, centrosomes and centrioles, nucleus, and microbodies. So I hope students are able to make a clear differentiation between the prokaryotic cells and eukaryotic cells based upon the cell organelles. So if we observe, this is the structure of the eukaryotic cell. So we have this is the structure having rough endoplasmic reticulum, and this is the nucleus which is present. This is the Golgi apparatus. This is the nuclear envelope, and this is the vacuole which is in between, vacuole, and this is the cell wall. This is the mitochondria. This is the microtubule. These are the plasmodesmatic bridges, these green color structures. We have rough endoplasmic reticulum, smooth endoplasmic reticulum. So like all the cell organelles, this is a detailed structure of all these cell organelles. Now let's discuss about each one. Coming to the cell membrane. So firstly, the chemical studies were conducted on human RBCs. So the main point which we have to remember here is cell membrane is a bilipid layer which is composed of proteins and carbohydrates. So this is the layer wherein we have protein here, and we have two molecules, carbohydrates, which are present in the form of sugar, and protein. So that is the reason here we have a combination of both. It is written as lipid bilayer. And yellow color structures which we have beneath, last in the bottom, these yellow colored structures are nothing but the cholesterol. So lipids mainly phosphoglycerides which have outer polar head and inner hydrophobic tails. So just as I have discussed about the permeability of cell membrane in the prokaryotic cells, here the lipids will have polar head outwards and inner hydrophobic tails. So any molecule will enter into the outside, but they are not permeable into the inner side. The reason is hydrophobicity does not allow them to enter into the inner membrane. Hydrophilic is water attracting and hydrophobic is water repelling. So they will not be able to enter. So that is the reason the non-polar tail of the saturated hydrocarbon is protected from this aqueous environment. Aqueous environment is nothing but the environment which has water. Now, if at all we observe the ratio of protein and lipid, it's generally different in different cells. For example, in human RBC, we have 52% protein and 40% lipids. So, based on the ease of extraction, again, they are of two types of proteins, integral proteins and peripheral proteins. Peripheral, the name itself indicates that they are lying on the surface. So, that is the reason periphery is nothing but outer side. So, that is the reason they are peripheral proteins. Partially or totally buried inside the membrane. So, that is the reason we call them as integral protein. We can remember the name of this protein by their as they are present only on the surface. So generally we call them as periphery. Periphery is nothing but outermost side. So remaining the next one is obviously opposite. Now this entire concept of cell membrane was proposed or based upon the fluid mosaic model of cell membrane, which was proposed by Singer and Nicholson. This was a very important question. It's most of the times asked in the exam. So Singer and Nicholson fluid mosaic model, it is a popular name of this model. The nature of the cell according to this model is like quasi-fluid nature, that is nothing but it enables the lateral movements of protein within the bilayer. So almost half of the membrane is permeable. So that is the reason we call it as quasi-fluid nature. Overall, the lipid bilayer, this layer. So that is nothing but the ability of the protein to move within this ability to move within the carrier protein for their transport is known as facilitated diffusion. So if at all you remember the concept of transport of plants in the first year, we have this term called facilitated diffusion, wherein only very few molecules will enter into the innermost membrane of the cell. So this process is known as diffusion, like movement from one area of the solute, lower area to higher area, that is through a membrane. It is known as facilitated diffusion. So this fluid mosaic model talks about the facilitated diffusion process mainly. Now see, if at all we observe again, how does this facilitated diffusion happen? This happens based upon the transport of molecules. Basically, it is again of two types, passive transport and active transport. So here if we observe, molecules are given about simple diffusion, facilitated diffusion, it's happening through a membrane. So it is clearly understood that molecules are transported through facilitated diffusion. And here is the ECF and ICF. If we consider two layers, as I have just now said, the movement of the molecules is from higher level to lower level. So here extracellular is one level and the intracellular is another level. So in between them we have the cell membrane. So this process of transport generally happens between the ECF and ICF. So if we observe, it is through these protein channels. Green colored one are the protein channels and these are the carrier proteins. So if it is not permeable, they cannot enter into the molecule. See, it is completely closed. If it is semi-permeable, they can enter and only few of them are entering into the intracellular space. These proteins are known as carrier proteins. So as it is selecting only few molecules, we call it as selectively permeable to some molecules present on either side. Due to this nature, plasma membrane helps in these processes like cell growth, formation of intracellular junctions, secretions, cell division, endocytosis and exocytosis. Endo is nothing but inside, exo is nothing but outside. Now here, as I discussed, there are two kinds of transport. One is passive transport and the other one is active transport. So the movement of molecules across the membrane along the concentration gradient, as I have said, if it is from higher concentration to lower concentration, here it is given simple diffusion is nothing but neutral solutes will move across the membrane, whereas osmosis is only about water by the diffusion across the membrane. And polar molecules are nothing but molecules having water that cannot pass through this non-bipolar lipid layer. So that is the reason they require a protein called as carrier protein. So these are those carrier proteins which help in the process. Hence this diffusion is referred as facilitated diffusion. This is about passive transport. Coming to active transport, here ATP and ADP are utilized. So that is the reason it is very clearly given, ADP and ATP. So here the movement always happens against the concentration gradient, that is the reverse of the facilitated diffusion, from lower to higher. Here it is from higher to lower, whereas it is from lower to higher concentration. Here it is without energy expenditure, here it is with energy expenditure. What is this energy? The energy here is ATP. So this is a brief overview of how the transport happens in the cell membrane, especially in a eukaryotic cell. Now moving on to cell wall structure. This is a non-living rigid structure and it is mostly found in fungi and plants. For example, in the cell wall of algae, it is made up of cellulose, galactones, mannans, and minerals like calcium carbonates. Whereas in other plants, it is made up of cellulose, hemicellulose, pectins, and proteins. So here we have primary cell wall, secondary cell wall. The cell wall is divided into two components. Primary cell wall is the outermost layer, and secondary cell wall is the innermost layer. So if this is the structure of cell wall, we have three layers like this: primary, secondary. So this is the first layer, this is the innermost layer. This enclosing is nothing but, here we have clearly, this is the middle lamella. So the cell wall and middle lamella have these bridges, green color bridges known as plasmodesmatal bridges. These are those bridges which help in the binding of the cytoplasm and the neighboring shells. So the important functions are like, it gives shape to the cell, it protects the cell from mechanical damage, helps in interaction with the other cells, and it also acts as a barrier for the undesirable molecules. Now moving on to the next most important part, that is endomembrane system. Endomembrane system majorly includes four important cell organelles within it. So if we observe the structure, of endomembrane system here, we have endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic reticulum. We also have, this is the Golgi apparatus, and we have lysosomes and vacuoles also present in the structure. So this is the rough endoplasmic reticulum, smooth endoplasmic reticulum, and this yellow colored structure is the vacuole. So endoplasmic reticulum is nothing but, these are tiny network tubular structures found inside the cell space. So it divides the cell inside the endoplasmic reticulum into two, cytoplasm also, as a rough endoplasmic reticulum and smooth endoplasmic reticulum. A rough endoplasmic, and smooth endoplasmic reticulum. So this is almost observed on the surface only. So here if we can see, it is present only on the surface here, whereas in the smooth endoplasmic reticulum is present inside these structure. So here it actively helps in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. So it is present out on the outer membrane of the nucleus, whereas it is present inside. Here we don't find the ribosomes, but we find ribosomes on the rough endoplasmic reticulum. So the clear differentiation between RER, rough endoplasmic reticulum, and smooth endoplasmic reticulum is through ribosomes only. Whereas a smooth endoplasmic is major site for synthesis of lipids, and in case of animal cells we even have the steroidal hormones synthesized in the smooth endoplasmic reticulum. So coming to the next part, the lysosomes. These are the membrane-bound organelles inside the nucleus. This is the very small structure. So this is about lysosomes, which are formed by the packaging in the Golgi apparatus. These are almost all types of hydrolytic enzymes present in them, like hydrolases, lipases, and all the things. They are very much active in case of acidic pH. They have this ability of digesting the carbohydrates, proteins, and all these nucleic acids also. Next, coming on to the vacuoles, the centralmost part present inside the nucleus internally is the vacuole. It almost has water, sap, all the excretory products present in them. It is a single membrane bound organelle which is known as tonoplast. In plant cells, almost 90% of the place is occupied by this vacuole only. And this tonoplast, which is present as a single layer, it helps against the movement and transport of ions against the concentration gradient. So it is a form of active transport here again, as discussed in the earlier slide. So the concentration here in the vacuole is always higher than compared to cytoplasm. So that is the reason, if at all, the best example if we observe in amoeba, contractile vacuole will help in the process of excretion. Moving on to Golgi apparatus, it is a stained reticular structure which is found in the nucleus. So we can observe this twisted structure of Golgi apparatus. It was first discovered by Camillo Golgi, so that is the reason it has got the name as Golgi apparatus. It is a flat, disc-shaped and sac-like structure. All these are sac-like structures. These sac-like structures are referred as cisternae. It is given here, and these are concentratedly arranged with the convex cis forming phase and mature trans forming phase. So cis and trans phases are totally different, but they are interconnected. So this is the cis phase and this is the trans phase. So this helps mainly in the process of packaging of vesicles, protein synthesis or by the ribosomes, which are present usually on the outer side, and the formation of glycoproteins and glycoproteins. Moving on to the next cell organelle, it is mitochondria. So as we all know, mitochondria is powerhouse of the cell. So the reason why we call it as powerhouse of the cell is it has a major form of energy production, that is the ATP. So about the mitochondria structure, so it is nothing but a double layered membrane bound structure with an outer membrane and inner membrane. It divides the lumen is nothing but the empty space, into two aqueous compartments, that is outer and inner compartments. Inner one is known as matrix. So here we have crystal like dots in between and these foldings. So the inner membrane, the matrix compartment, has certain infoldings. So these infoldings of the inner matrix are referred as cristae. So these inner foldings, are referred as cristae. So these generally increase the surface area of the mitochondria. So the two membranes have their own specific enzymes associated with the mitochondrial function. So basically the matrix here, this green colored structure, basically the matrix is nothing but the gap present in between the cristae, the foldings. So these have another structure called circular DNA enclosed within it. and along with that few RNA molecules, ribosomes, and certain components which are needed for the protein synthesis. So the mitochondria generally divide by fission. Fission is nothing but it is talking about the reproductive process. So the major function, as I have just now said, it is the powerhouse of the cell. The reason is it acts as a site for aerobic respiration and it produces the energy in the form of ATP. Now ATP again will have ADP plus PI. ATP so ATP is broken into ADP plus PI or IP, both are the same. So ATP is adenosine triphosphate and ADP is adenosine diphosphate. Here IP or PI is nothing but inorganic phosphate. So if you observe the structure of mitochondria carefully, this is the outer membrane, this is the inner membrane, this is the intermembrane space present in between those two, and these enfoldings, as I've just explained, are known as cristae, and we have a portion in between them called as F0 portion and F1 portion, and in between the cristae we have our DNA present and granule-like structures. These green color dots are granule-like structures known as ribosomes. Moving on to the next part of eukaryotic cells, we have another cell organelle known as plastids. So plastids are nothing but they are commonly found in all kinds of plant cells and in nucleoids. They are large sized or small sized, but they are easily observable under the microscope. They generally contain some pigments. So based upon the type of pigments present, these plastids are basically classified into three types, that is chloroplasts, chromoplasts and leucoplasts. So if they contain a pigment called chlorophyll in them, then they are known as chloroplasts, and carotenoid pigments also. So if it is a combination of chlorophyll and carotenoids, then we call that plastid as chloroplast, which is helpful in the movement, that is trapping of light energy for photosynthesis. Next comes the chromoplasts. These contain fat soluble carotenoid pigments like keratin and xanthophylls. So this gives a yellow and orange or a red color. Basically all yellow and orange colored vegetables and fruits will have this chromoplasts. And next coming to leucoplasts. Leucoplasts are popularly colorless plastids which have various sizes and shapes. They store nutrients in them. So we find leucoplasts again in three major forms, such as amyloplasts which store starch, for example potato, and elaioplasts which store oils and fats, and aleuroplasts which store proteins. So in case of rice structure, if we observe the bran internal structure, we can find these aleuroplasts where they generally store these proteins. So if at all we observe this picture, we can clearly identify. So this is a chloroplast. Leucoplast is generally colorless. Elaioplasts store oils and fats, so that is the reason these are the oil molecules, and amyloplasts are having starch in them. The starch is nothing but mainly found in potatoes. Next, these are remaining different kinds apart from these three: etioplast, proplastids and proteinoplasts. Now discussing in detail about chloroplasts, which is having chlorophyll and carotenoid pigments in them. So this is the microscopic view. This is the microscopic view of chloroplast, and here we will have the longitudinal section of chloroplast, wherein we have the thylakoid granules, intermembrane space, outer membrane, inner membrane, and this is the transverse section of chloroplast. So it is a double membrane cell organelles which include all mesophyll cells in various shapes like oval, spherical, discoid and ribbon-like. It is less permeable the inner membrane is less permeable to any kinds of solutes or solvents, and the space is limited by inner membrane of the chloroplast. This space is known as stroma. This stroma, if you observe the structure carefully, here we have these stroma. These structures are known as stroma. So they contain small double-stranded circular DNA molecules, ribosomes, enzymes, which are required for the synthesis of carbs and proteins. So inside we have these flattened green color sac-like structures are known as thylakoids, which are present in the chloroplast pigments. These are arranged in the form of stacks, and these stacks, whenever they are piled together, that area or that region is known as grana or intergranal thylakoids, and they have a flat membranous tubules in between them connecting to each other. They are known as stroma lamellae. That is what is given here, stroma lamellae, which are connecting the thylakoids of different grana, and the space present in them, as already said, that is lumen. Here also it has two kinds of ribosomal units, which is 70S and 80S, present in the chloroplast other than the cytoplasmic ribosomes. Moving on to the next cell organelle, it is the ribosomes. So ribosomes here have two kinds of subunits, wherein 60S is the larger subunit, 40S is the small subunit. Together they form 80S. 80S. So S here is the sedimentation coefficient. So if you observe the figure here, this is the smaller subunit, this is the larger subunit. Together they form 80S ribosome. So within the cell structure, if we observe the Golgi apparatus, here we have certain granules moving freely inside the cell structure near the Golgi apparatus. That freely moving structures, these granule-like structures, are known as ribosomes, which are always associated with rough endoplasmic reticulum. So these are known as free ribosomes. So the presence of ribosome is only found in rough endoplasmic reticulum, that is RER, whereas smooth endoplasmic reticulum do not have these ribosomes. Moving on to the next structure, that is the cytoskeleton. Cytoskeleton is nothing but it is the outermost part here, if we observe in this picture, which is a network of filaments like proteinaceous structures present in the cytoplasm. Basically, main function of this cytoskeleton is to provide mechanical support, motility, that is nothing but movement, and maintaining the shape of the cell. So here if we observe, these tube-like structures are nothing but the filamentous proteinaceous structures which are present inside the cytoskeleton here, and these three are the major important functions, that is support, movement, motility is nothing but movement, and maintaining the shape of the cell. Moving on to the next cell organelles, we have cilia and flagella. So cilia and flagella are nothing but hair-like outgrowths of the cell membrane. So if at all we take the structure of the cell like this, we have these structures coming like hair-like structures. This is the flagella, and these hair-like structures are the cilia. So cilia are nothing but small hair-like structures which work like oars, is nothing but they cause the movement of cell in the surrounding fluids. Whereas flagella, they are longer, as I have said here, they are longer like this, usually in ribbon shield, which are responsible for the cell movement. So the flagella are generally different in eukaryotes and prokaryotes. They are generally covered with plasma membrane, and here if we observe the internal structure of flagella, we have the popular arrangement known as 9+2 array. That is nothing but here this red color structure is known as the axonome, which represents the core, which has many microtubules. These blue color structures are microtubules, which are arranged 9 pairs of doublets radially arranged in peripheral microtubules and a pair of central microtubules. So these are totally 9 in number, and we have a pair of central microtubules, 2 in number. So this is the reason we call it as 9+2 array. So flagella are mostly found in this format of 9+2 array. Here the central tubules are connected by bridges and they are enclosed by central sheath, and they have 9 radial spokes and the peripheral doublets. So if you observe, there are 9 doublets here. These doublets are again linked to each other within the circle. They are linked to each other with the help of linkers. That is nothing but they are interconnected to each other with the help of linkers. So basically the main function of cilium and flagellum is like they help in the motility movement of the bacteria. Moving on to the next structure, that is centrosomes and centrioles. These are two non-membrane bound cylindrical structures. If we observe in the figure, these are cylindrical structures like this, which together are always present in the form of pairs, and these centrioles lie perpendicular to each other, and they are made up of 9 evenly spaced peripheral fibrils of tubulin. If at all we observe, tubulin is nothing but the protein that is present inside these centrioles. Here we have a central part of the centriole called as proteinaceous hub, which are connected again by peripheral triplets by radial spoke of proteins known as tubulin. So here if we observe, these are the tubule-like structures known as microtubules. They are connected by a radial hub of proteins. That protein name is like tubulin. This central part is the centriole. So here the centrioles again form the basal body for the flagella and help during the mitotic phase of cell division. They act like spindle fibers and spindle apparatus usually found in the cell division during the animal cell. So these centromes and centrioles help in the reproduction process, that is as appearing themselves in the form of spindle apparatus during the mitotic division process. Moving on to the next most important cell organelle, that is the nucleus. So the major difference between eukaryotes and prokaryotes is with the help of nucleus. So prokaryotes may sometimes have the nucleus, or they may not have the nucleus. False nucleus may be present. But in case of eukaryotes, means true. So always eukaryotes are always having nucleus enclosed within them. So nucleus was first discovered by Robert Brown in the year 1831. So this was found by staining it with a basic dyes in the name of crystal violet blue. So a part of material of nucleus was stained by these basic dyes, and it was given the name of chromatin. So this experiment was carried out by Sir Fleming. So in this way, this nucleus was discovered. Normally, every cell has one nucleus, but some cells may have more than one. Some mature cells do not have nucleus. For example, in humans we have mammalian RBC, sieve tubule cells of vascular plants. They have nucleus. So interphase, generally we have prophase, metaphase, 4 phases are there in the process of cell division. So whenever we are concerned with interphase of nucleus, generally 4 parts are developed within the nucleus. That is the nuclear envelope, nuclear matrix, chromatin and nucleolus. So if this is the structure of nucleus, if we observe the structure of nucleus here, the outermost part here in which the nucleus is enclosed is known as nuclear envelope, and this structure which is present in between is the nucleolus, and the protein parts which are connecting that to the nucleolus is the chromatin, and the remaining dot-like structures are the endoplasmic reticulum having the ribosomes, and the pores which are present here are the nuclear pores. So this is basically the description of nuclear envelope. It is a double membrane space between perinuclear space which is present in the nucleus. Next we have the nuclear matrix, which is also known as nucleoplasm, which is simply a fluid which has nucleus within it, and it has various materials such as chromatin and nucleolus, which are helpful for the nuclear functions. This nuclear matrix or nuclear envelope or the nucleus, all these organelles which are enclosed within it play an important role during the cell division. So that is the reason this nuclear matrix is also known as karyolymph or nuclear sac. And we have a chromatin in the form of a nuclear protein, which is a network of nuclear proteins which has DNA proteins, which carries the genetic material in the form of DNA, and it has two kinds of proteins, that is basic proteins, the histones, and non-histone proteins called as RNA. This chromatin plays an important role during the cell division because this chromatin will condense to form the chromosomes. So that is the reason nucleus plays an important role during the cell division in the form of chromatin networks like spindle fiber formation as discussed in the previous slide. Next, coming to the last important part is the nucleolus. Generally, there may be one or more than non-membranous spherical bodies, the nucleolus, which is generally continuous with the nucleoplasm, and it is important for the site of ribosomal RNA synthesis. This is in place of translation, we have this ribosomal RNA synthesis. Translation, we have this ribosomal RNA synthesis. In that place, this nucleolus plays a major role. So this is all about the most important cell organelle, the nucleolus. Now moving on to the next is cell organelle microbodies. The name itself indicates they are very much small in their structure. But they are very much minute, present in the form of vesicles which contain certain enzymes, but their presence can be found in both plant and animal cells. Now, if at all we observe the eukaryotic cell structure in the plant and animal, we can notice major differences here. So the differences are in the form like, in the plant structure we have endoplasmic reticulum is one characteristic feature which we can differentiate. Both of them are having endoplasmic reticulum, but the functions vary within each of them. And we can also make out the plant cell with the help of vacuole, whereas vacuole is generally not present in the animal cell. Now let's discuss the detailed difference between prokaryotic and eukaryotic cells. So in case of occurrence, prokaryotic cells are generally found in blue-green algae, whereas eukaryotic cells are found in animals, plants, algae, bacteria, almost in all kinds of living organisms. Coming to the size, here we have prokaryotic cells are usually from 1 to 10 micrometers, whereas eukaryotic cells are from 10 to 100 micrometers. And the occurrence of prokaryotic cells in the form of multicellular organisms is very rare, but in eukaryotic cells we have it in the form of extensive tissue formation. Coming to cell wall formation in prokaryotic cell, it is present in most of the cell but not in the form of cells. That means, for example, if we consider cyanobacteria, here the bacterial cell wall is made up of murin polysaccharides, lipids and proteins. So apart from its presence as a cell wall, it is present in the form of either protein molecules or lipid molecules or starch molecules. That is the characteristic of cell wall pertaining to prokaryotic cell. Whereas in eukaryotic cell, in animal cells basically they lack cell wall, but the plant cell is made up of cell wall and chitinous cell wall which is present in the fungi. So chitin is a protein which is basically present in the fungi, that is having eukaryotic cell. We have the presence of plasma membrane in prokaryotic cell as well as eukaryotic cell. The major difference, as I have discussed in the previous slides, related to nucleus. Nuclear membrane is absent in prokaryotic cell but present in eukaryotic cell. In the same way, chromatin with histone proteins is absent in prokaryotic cell and it's present in eukaryotic cell. In the terms of number of chromosomes, here only each cell has one chromosome in prokaryotic cell, but the number of chromosomes per cell depend upon the type of organism when it comes to eukaryotic cell. Moving on to the next feature, the chromosome here in the prokaryotic cell is in the form of circular ring lacking a centromere. So best example is in case of a bacterial cell, we have this circular ring in the form of plasmids. In the form of plasmids, we can find this chromosome, but not in any kind of not in any kind of cell material related. They are present as plasmid rings. But here in the eukaryotic cell, we have the chromosome in the form of a linear format, and it has a centromere and centrioles involved in it. Coming to the most important part, the genetic material. So here in prokaryotic cell, we only can find double-stranded DNA either in the circular format or as a linear form, which have exons present in them. Whereas in eukaryotic cells, we observe linear double-stranded DNA genes frequently interrupted by intron sequences, in case of split genes. They are usually present as intron and exon sequences in case of eukaryotic cells. But here in prokaryote, their occurrence is only like double-stranded DNA. The nuclei and mitotic apparatus are absent in prokaryotic cell and they are present in eukaryotic cell. That is the reason in the nucleus cell organelle we have discussed in detail at how these nuclei and mitotic apparatus, the chromatin disappearance and appearance, how it happens. Coming to nucleolus, it is absent in prokaryotic cell and it is present in eukaryotic cell as well. It helps in the process of organization and synthesis of ribosomes in the eukaryotic cell. So as I've just now said here, plasmid concept. The plasmids are commonly present in prokaryotic cell, but their occurrence is very much random in case of eukaryotic cell. And the presence of mesosomes. Mesosomes are present in prokaryotic cells. In the second cell organelle of prokaryotic cell, I have discussed about mesosomes and chromatophores. So mesosomes are present in prokaryotic cells, which are majorly helping in the functions of Golgi bodies, mitochondria, and they help in the separation of chromosomes during cell division. So mesosomes help in this cell division process in terms of prokaryotic cell. Whereas in eukaryotic cell, we can observe nuclei and mitotic apparatus and nucleolus, especially the chromatin and all the internal organs which are present in the nucleus. So that is the reason mesosomes are absent in the eukaryotic cells. Moving on to the next feature, this is a detailed description related to in the form of a comparison in the form of a tabular form of cell organelles, wherein the mitochondria are absent in prokaryotic cell where it is present in eukaryotic cell. In the same way, vacuoles are absent in prokaryotic cell and it is present in eukaryotic cell. Because in every plant cell we can find vacuole which they act as a reserve for the food materials and storage of food, and they help in the utilization of it in the form of energy in adverse conditions. Coming to lysosomes, lysosomes are present only in prokaryotic cells and they are absent in eukaryotic cells. And chloroplasts are absent in prokaryotic cells and are present in eukaryotic cells because we have plastids. So in that form, as discussed, there are many forms of storage plastids. Chloroplast is one of the major form in which eukaryotic cells have it. Next, coming to centrioles. Centrioles are absent in prokaryotic cells and are present in eukaryotic cells because the cell division process is carried out with the help of centrosomes and centrioles only. Coming to ribosomes, here in prokaryotic cell we have only 70S type of ribosomes, whereas in eukaryotic cells we have 80S in the form of ribosomes, and the plastid and mitochondria also have 70S ribosomes. So both kind of ribosomes structures are present in eukaryotes. Only 70S is present in prokaryotic cell. So microtubules, these are the cilia and flagella things these two are talking about cilia and flagella arrangement, that is about the 9+2 arrangement which is given here. So it is absent in case of prokaryotic cell, present in eukaryotic cell. Flagella has the flagellin material in prokaryotic cell. There is no 9+2 structure found in the prokaryotic cell, but whereas in eukaryotic cell we can find this 9+2 structure along with tubulin and proteinine proteins, whereas flagellin is a protein in prokaryotic cells. Coming to respiration, respiration is strictly by anaerobic process, so that is the reason it is given as many strict anaerobes. Here it may be by facultative anaerobic process, or most of the times it's by secondary modifications, like we have mycorrhizae, fungal roots modification, or else pneumatophores which are breathing roots. So in eukaryotes, as they are plants having several kind of modification, we are having facultative anaerobic process. Coming to photosynthetic enzymes, they are bound to plasma membrane as a composite chromatophore in case of prokaryotic cell, whereas in eukaryotic cell, the enzymes are packed in the form of plastids which are bound by membrane. So there comes our example of chromoplast, chloroplast, alluroplast, all these kinds of storage plastids. Moving on to the metabolic patterns, in prokaryotic cells there are great variations, but in eukaryotic cells we have all these plant cycles, C3 cycle, C4 cycle occurring in the cytoplasm, electron transport chain, Krebs cycle, oxidation, glycolysis, several kind of metabolic patterns are found in eukaryotic cell. But in prokaryotic cell we cannot find those kind of patterns. Coming to the sexual system, in prokaryotic cell it is very rare, like we only have one way, either it is by haploidy or diploidy, or by the formation of merozygotes, but only one parent is involved here. But here both the sexes are involved in case of eukaryotics in the sexual participation, and the entire genomes are transferred in this process. Coming to cytosis, there are no streaming events of cytoplasm in prokaryotic cell. That is, the cytoplasm here is constant and there is no movement in terms of cilia or flagella. But here in case of eukaryotic cell, cytoplasm shows streaming movements. So best example for this is amoeba. So amoeba, we have the excretion in the form of contractile vacuoles. So we can take this cytoplasm streaming movements example in the eukaryotic cell. Coming to protein synthesis, here in prokaryotic cell we have transcription and translation taking place in the cytoplasm. So both transcription and translation occurs in cytoplasm only. But in eukaryotic cell we have transcription in nucleus happening and translation taking place in cytoplasm only. So there are two different cell organs in which transcription and translation both take place in two different cell organelles when coming to eukaryotes, but it happens only in cytoplasm in prokaryotes. Coming to the most important part of the duration of cell cycle, here we have noticed that there is a short cell cycle in case of prokaryotes, wherein it takes only 20 to 60 minutes to complete, whereas the cell cycle is very long in case of eukaryotic cells, that it takes 12 to 24 hours to complete the cell cycle. I hope we are clear with the topic about the difference between the prokaryotic and eukaryotic cells. Almost 20 to 25 points of feature differentiation, characterize differentiation between eukaryotes and prokaryotes were discussed in detail. Thank you, one and all.